Hello everyone, my name is Gary O'Mealy, and in this video we are going to talk about protein synthesis. So we've spent several videos in this video series discussing amino acid, protein structure and function, and specifically how proteins fold into functional proteins that are going to aid the cell in maintaining normal cellular activities, and then we've kind of looked at how that is going to impact physiological activity at kind of a higher level. So we're also going to want to make sure that we're aware of how proteins are actually synthesized inside the cell. We haven't really talked about that in any sort of detail yet. So both the process of forming peptide bonds between amino acids, building proteins and folding proteins, these are processes that are mediated by machinery inside the cell, and these processes tend to be very well regulated and can even be directed by physiological processes, as we will see. So when you're talking about the step-by-step method in which proteins get made, you're really talking about what's called the central dogma of molecular biology. This central dogma describes all the molecular processes that the cell will go through in order to synthesize proteins. So it's all going to start at the DNA level. So a gene, you've probably heard this term before, a gene is a coding segment of a chromosome that provides the instructions for making one specific protein. So there are roughly 30,000, give or take, genes that are found in the human genome across all 23 of our chromosomes. The idea is that each one of those genes provides the blueprints on how to make one protein, okay? So I mentioned that we have 23 unique types of chromosomes, 46 in total because we've got 2 copies of each, one from mom and one from dad. Across all 23 chromosomes, there are roughly 30,000 genes, and these chromosomes are found inside of the cell's nucleus. So the first step in making a protein is transcription. So during transcription, an enzyme which is in the nucleus, this enzyme is called RNA polymerase, it works with a number of other proteins in the nucleus to synthesize a messenger RNA transcript that contains the exact same information as one particular gene. You could think of this mRNA transcript as being a photocopy of the instructions for making one protein. So in this illustration, you can see that a lot of what goes into the transcription process is getting access to the DNA, getting access to the one gene for that one protein that we want to make, unwinding the DNA, and then RNA polymerase will synthesize an RNA transcript that is complementary to what is in the gene of the DNA. So in addition to RNA polymerase, we also want to introduce another important protein that is involved in transcription, and these proteins are called transcription factors. They tend to bind to the DNA in regions that are upstream of the coding or information-containing region of the gene, and this region is called the promoter. So the coding region, sometimes also called an open reading frame, contains the exact information on how to build the protein. The promoter upstream here is a lot more regulatory in nature. This is going to contain the sequences that will recruit RNA polymerase to this particular gene so that we can synthesize this particular protein at the right time and at the right place. So various transcription factors, which again are DNA binding proteins, once they are assembled at the promoter, they are able to recruit RNA polymerase to the promoter, and at that point, RNA polymerase can work to unwind the DNA and start synthesizing an mRNA transcript. To inject a little bit of physiological relevance into this, look at this scenario that we have here. We have a capillary on the far left, and in that capillary we have a circulating steroid hormone. We haven't really gotten to our chapter on the endocrine system yet, or how hormones do what they do and what hormones do, but this is so interesting I want to go ahead and introduce it now. In a lot of situations, the cell will constantly be synthesizing proteins that are needed all the time. So these proteins that are synthesized around the clock because they're needed all the time, they are sometimes called housekeeping proteins. Other proteins are not needed all the time. So for those proteins that are only needed in very specific situations, the cell has ways of inducing their transcription and translation in response to certain physiological stimuli. So in this particular case, we have a transcription factor that is floating around in this effector cell's cytoplasm, and it is in an inactive state. You'll notice that it is not in the nucleus, and therefore does not have access to the DNA. Well, one thing that we can do here is that this steroid hormone, which is being carried through the blood, it can diffuse out of the bloodstream into this effector cell, and oftentimes these transcription factors are what we call steroid hormone receptors. So the steroid hormone will bind to this transcription factor, and this actually turns this into an active transcription factor. This active transcription factor can now move into the nucleus, where it can find the promoter of whichever gene it is meant to activate, and that will eventually lead to the transcription of that gene into a messenger RNA, and then the eventual production of a protein that this particular cell was not making before. So, in essence here, the steroid hormone that we started out with here exudes its effect on this effector cell. It elicits a physiological response by getting this effector cell to make a protein that it was not making before. So any change in physiology that is going to occur here is going to be because of the function of this new protein. So we will see plenty of examples of that in the future. Okay, so moving on, after transcription occurs, the messenger RNA is in an immature state. So we're not really going to make a big deal out of it, so we're not going to talk about it too much, but there is some RNA processing that goes on inside the nucleus. We have to remove the non-coding introns and splice the coding exons together. So once this is done, the mature messenger RNA is exported out of the nucleus and into the cytoplasm. From here, the messenger RNA will be bound by a ribosome, which we understand now is a protein-building machine. The ribosome can either exist freely in the cytoplasm, or these ribosomes can be embedded in the rough endoplasmic reticulum. And which one of these ribosomes the messenger RNA binds to oftentimes is determined by whether the protein we're going to make is a freely soluble protein or a membrane protein. Membrane proteins, integral membrane proteins, tend to be synthesized in the rough ER. So the ribosome, which is our protein builder here, it is essentially going to read the blueprints for how to make this protein, which is the messenger RNA. The ribosome will scan the blueprints, the messenger RNA, from 5' to 3', searching for the start codon, the signal that says this is where the instructions begin, this is how you start building the protein. So the start codon is AUG. So the ribosome, once it binds to the far 5' end of the messenger RNA, is going to scan through the code in the messenger RNA, looking for the first instance of AUG. So once this start codon is encountered, transfer RNAs are going to begin delivering particular amino acids to the ribosome, and which amino acids they are bringing in is, of course, dictated by the genetic code. So you can actually see the genetic code here on the right side of the screen. Each codon, or sequence of 3 RNA nucleotides, codes for a different amino acid. There are 64 different codons, but only 20 amino acids to code for, so you'll notice that there are a lot of redundancies here. For example, all 4 of these codons here code for the same amino acid alanine. All 4 of these codons here code for the same amino acid valine. Importantly, though, you're not going to need to memorize the genetic code, but there are a few things that you will need to remember. First, AUG is the start codon, and that always codes for the amino acid methionine. This explains why most proteins start with methionine. The only proteins that do not start with methionine are the ones that go through some cleaving, and the beginning of the protein must have been chopped off at some point. So each codon that comes after the initial start codon, which again is methionine, will code for one of the other amino acids that are going to need to be incorporated into the protein. The very first one, methionine, will be found at the N-terminus of the protein, and whichever amino acid gets incorporated in last is going to be at the C-terminus. So eventually, as the ribosome scans the messenger RNA, it will encounter a stop codon. Your stop codons are UAA, UAG, and UGA. If you ever have a hard time remembering the stop codons, here's a little trick. Think of somebody that you really don't like. I'm talking you really don't like this person. The stop codons can represent things that you might say to this person. UAA, you are annoying. UAG, you are gross. And UGA, you go away. So that's kind of a fun way of remembering the stop codons. So there are 3 stop codons, and each one, when the ribosome encounters it, will indicate to the ribosome that protein synthesis has been completed, and it's time to let go of the protein so that it can start folding into its fully functional shape. So here you can see an illustration of how the transfer RNAs are bringing in their respective amino acids. Here's your start codon here, and you can see this initial transfer RNA is bringing in a methionine amino acid. So the large subunit of the ribosome contains little cavities that will accommodate the transfer RNAs and the amino acids, so that we position our initial transfer RNA with its methionine here, and then the next codon over is going to code for phenylalanine. Another tRNA brings that in here, and then at this point it is the ribosome's job to join those 2 amino acids together by dehydration synthesis. The ribosome, in particular the ribosomal RNA of the ribosome, contains the peptidyltransferase enzyme activity that is necessary to forge these peptide bonds. And basically what the ribosome is going to do here is continue to scan through the messenger RNA, bring in new transfer RNAs, bring in new amino acids, and continue to chain together and build this actively synthesized protein, 1 amino acid at a time. So here would be the N-terminus of the protein, and we continue to build our protein from N-terminus to C-terminus. So again, as the ribosome scans this messenger RNA 3 nucleotides at a time, it's going to continue to forge peptide bonds over and over and over again. It's going to continue to build a amino acid polymer, a polypeptide, and the sequence of amino acids in this polypeptide should exactly match what is dictated in the genetic code, in the DNA code. The DNA provides the instructions on how to build the protein, and the way to build a protein properly is attach the amino acids together in the proper order. We talked in a previous video about the importance of primary sequence in determining the folded structure of a protein, and therefore what the function of that protein is going to be. So in this illustration here, you can kind of see all this put together. Here is the double-stranded DNA. When we peel it apart and synthesize a complementary messenger RNA, you'll notice that this messenger RNA contains exactly the same information in the coding strand of this DNA here, ATG-CCG, AUG-CCG. So the only difference between this messenger RNA and the coding strand of the DNA is the thymines in the DNA have been replaced with uracils in the RNA. Otherwise, the information is all the same. And then you can see during the translation process, when the ribosome is reading the instructions in the messenger RNA, each codon or triplet of nucleotides codes for one particular amino acid. So for this protein that we're building here off of the genetic instructions, from N-terminus to C-terminus, the sequence of this protein would be methionine, proline, glutamine, serine, valine, histidine, alanine, leucine, methionine, and then cysteine, from N-terminus to C-terminus. Okay, let's wrap up this video by talking a little bit about genetic mutations. Mutations are something that gets a very bad rap, or at least gets way exaggerated in terms of their significance. in kind of a sci-fi context, you think of mutation, you're thinking of like some hideous monster that has tentacles for sprouting out of its face, has 20 arms and 20 legs. That's not what a mutation is. A mutation is just a change in the DNA code. If you pulled out a book off of your bookshelf and you started taking a pencil and marking out words and then replacing them with new words, that's exactly what a mutation is. You are just changing the information that is in the genetic code from one thing and you're changing it to another thing. So if the mutation occurs in the coding region of the gene or the open reading frame, then what we're going to end up doing possibly is changing the amino acid sequence of the coded protein. If you start swapping around the order of amino acids in the primary sequence of a protein, you can end up having very, very, very significant effects on protein folding and protein function. So there are all different types of mutations. Some mutations are very, very large scale because they involve the addition or deletion of large stretches of DNA. We're going to focus on much more direct mutations. These are called point mutations. A point mutation is a change of one nucleotide in the DNA code. So here, we're going to first consider the wild type version of the DNA. Wild type in this case refers to the non-mutated version of the DNA, or the DNA or allele that is found in the majority of the population of whatever species you're talking about. So let's say the wild type version of this particular codon is TTC. So when we make messenger RNA off of the template strand, the messenger RNA will be complementary, so it will be AAG. Okay, so if you look at AAG in the genetic code, that codes for the amino acid lysine. So when the ribosome encodes AAG in a messenger RNA, lysine will be incorporated into the growing protein. So let's look at our first kind of point mutation. This is called a silent mutation, and you'll see why here in a second. So let's say that we change this C here into another T. So the messenger RNA will be AAA, but if you look at the genetic code, AAA also codes for lysine. We mentioned that there were redundancies in the genetic code, and this is a perfect example of that. So even though we have changed the nature of the DNA and the messenger RNA, because we have not changed which amino acid has been incorporated into the protein, we call this a silent mutation because there will be no noticeable effect on protein folding and protein function. You're sticking the same amino acid in you would have anyway, so the protein is not going to really change at all. Next, let's look at a much more serious mutation. This is called a nonsense mutation. So here, we take the wild type codon and we replace the first T with an A. This is going to result in the messenger RNA codon being UAG, which you will recall is a stop codon. This can be absolutely disastrous for a protein depending on where this mutation is found. If this mutation is found near the beginning of the coding region for that particular gene, then the protein that ends up being made is going to be much, 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 much shorter than it should be. So if a protein is supposed to be 1,000 amino acids long, a nonsense mutation that occurs very early on in the reading frame may cause this mutated protein to maybe only be like 50 amino acids long. So a truncated or extra short protein in almost every case is going to be incapable of doing its job. So of all the mutations we're going to talk about here, the nonsense mutation is possibly the most deadly or most potentially deadly. Okay, so the next mutations we're going to talk about are called missense mutation. These are a little hit or miss depending on based on kind of how serious they end up being for protein function. So there are two types of missense mutations that we're going to introduce separately here. Let's look at the left one here. We have changed the middle nucleotide of this codon from T to C. So in the messenger RNA, this codon will now be AGG. This amino acid will now be arginine. There is no question that arginine is not the same as the wild-type lysine, but you'll recall that arginine and lysine both belong to the same class of amino acids. They are both basic, and they are both positively charged. So the idea here is that the protein is going to be a little bit different because we have incorporated a different amino acid, but because we have incorporated an amino acid with very similar chemical properties, the hope is that maybe protein folding and function is not going to be affected all that much because we've replaced one positively charged amino acid with a different positively charged amino acid. So the hope here is that this isn't going to negatively impact things too much, but it still could because arginine is still different than lysine. So this type of mutation in which you replace one amino acid with a different amino acid that has the same chemical properties, this is called a conservative missense mutation. On the other hand, if we look at the other type of missense mutation where we replace the first nucleotide here with a C, this is going to lead at the messenger RNA level to GAG, which produces the amino acid glutamate, which is definitely different than lysine because glutamate is an acidic amino acid. It has a negative charge. It could not possibly be any more different than lysine. So this is an example of a non-conservative missense mutation. So the idea here is that we have replaced lysine, a positively charged basic amino acid, with glutamate, a acidic, negatively charged amino acid. We've replaced the amino acid here with one that has completely different chemical properties, and in all likelihood, this is going to mess with the way that the protein folds and the way that the protein functions. So to kind of tie this all together into physiology, genetic diseases are heritable diseases that can be traced to a harmful mutation that is passed on through generations. So really what we're talking about here are mutations that impact protein function and folding, and these proteins are going to have important functions in physiological processes. So if we mutate a protein and that protein no longer does its job properly, whatever physiological process that protein participates in, that process is going to suffer a great deal. So some examples of these that we'll talk about throughout the semester: sickle cell anemia is caused by a non-conservative missense mutation in the beta globin gene. Beta globin is one of the proteins that makes up hemoglobin, and the non-conservative mutation that's happening here is a glutamate, which is negatively charged, is turned into a valine, which is nonpolar and hydrophobic. In cystic fibrosis, this is the result of a nonsense mutation in a gene called CFTR. CFTR is a chloride transporter in the membrane of the digestive tract and the lungs. So the idea here is that a nonsense mutation will cause this CFTR protein to be way too short so that it does not do its job properly in transporting chloride ions, and this results in a lot of problems with water retention and fluid secretion in both the lungs and the digestive tract. And finally, in a much more broad sense, a lot of cancers are caused by mutations to genes that regulate the cell cycle, and we'll talk about a little bit of that in the next video. But the good news here is that random mutations that have been acquired in what are called somatic cells, which are most of the cells in your body, are not heritable if the mutation occurs in those somatic cells. The only way that cancer can really be passed on from generation to generation is if those mutations are acquired in what we call the germ line, the cells that are actually producing sperm and egg. Okay, that's going to do it for this video. Here is a list of vocabulary terms you probably ought to be aware of. And for checking your understanding, number 1, how is the nucleotide sequence of DNA related to the amino acid sequence of a protein? Number 2, how can control centers in the body direct effector cells to produce new proteins? And number 3, how can mutations to DNA affect normal protein function? Okay, that's going to do it for this video. If you have any questions, go ahead.